to be back. So thank you very much for that. Given the audience, I'm sure I don't have to introduce nematodes as very damaging pests. So it starts straight away. So in my PhD, I look a little bit at the nematode side, and I want to understand more about how the nematode parasitizes the plant and regulates its parasitism to then use it against the nematode. And key to nematode parasitism are small molecules termed effectors that the nematodes secrete into the plant to manipulate it. They have very important functions. For example, they help in the degradation of plant cell walls, which is important during invasion. They can also manipulate plant development. And last but not least, they're also very important during the suppression of plant immunity. So as you can see from all of these functions, these nematodes heavily rely on effectors to parasitize the plants. And over the recent years, we learned a little bit more about what these effectors do. But what we still know barely anything about is how effectors are produced. But that's important, because you can imagine if we can find some central regulator inside the nematode that regulates the production of a bunch of these effectors, we could try to target that regulator, and by doing so, target a lot of effectors at the same time and take away the key tools that the nematodes need to parasitize the plant. So how is effector production regulated? Most of these effectors become activated or expressed or maximally expressed when the nematode is already in contact with its host. So we started with this hypothesis in mind that maybe a regulator of effector production might respond to some kind of plant signal. And to test this hypothesis, we grew host plants in water. Our host plant here is mustard. And we then collected the water, the diffusate, which contains molecules outside the host root that the nematode might respond to. And we also cut off the roots, mashed them up to get signals inside the host root that the nematode might respond to. And then we treated juveniles of the Beetzus nematode, Heteroderishachtii, with either diffusate or extract. And we wanted to know what happens to gene expression in the nematode. And we sequenced this, and we found over 600 genes to be differentially expressed. We were able to cluster them into these 6 clusters that you see here, based on the expression, whether the expression goes up or down in contact with diffusate or extract. So this had profound influences on gene expression, and I want to take a closer look at today at this cluster of genes that becomes upregulated when the nematode is in contact with the root extract, so signals inside the host root. And what makes this cluster so interesting is that there are actually 60 differentially expressed effectors in this cluster. So this strengthened our hypothesis from the beginning that maybe effector gene expression depends on some kind of plant signal. But like I said, we are not interested in the effectors themselves. We're interested in regulators of effectors. So we also looked for transcription factors in this cluster, and we found 7. So now we had 7 candidates that might be regulators of effector gene expression. And we wanted to narrow down this list further. And for that, we didn't just take into account when effectors are expressed, we also took into account where effectors are expressed. As most of you know, effectors are predominantly produced in specific tissues, the gland cells. There are 2 subventral gland cells, which are more active during the earlier parasitic stages. And then there's the one dorsal gland cell, which becomes more active later on in the nematode life cycle. And now if we think about it, it would only make sense that a regulator of effector production is also highly expressed in these effector-producing tissues. So we wanted to look at spatial expression. And on the next slide, I'm going to show you the results for the candidate transcription factor that we chose to focus on. And we looked at spatial expression with a method called fluorescence in situ hybridization chain reaction. What you see in this picture here is a picture of a nematode. In the upper panel, you can see in cyan the transcript of our candidate regulator that we're investigating. Below that, you can see 2 controls. There's a dorsal gland control in yellow, and that's for a gene that we know is expressed in the dorsal gland cell. And you can see a subventral gland control in magenta, and that's for a gene that we know is expressed in the subventral gland cell. And now we can compare these 2 panels. And if we do this, we notice that the transcription factor that we're investigating in cyan nicely matches our subventral gland control in magenta. So this tells us that indeed this candidate regulator is specifically expressed in a major effector-producing tissue, especially in a tissue that's important early on during the nematode life stages. So we now were more confident in this regulator, and we wanted to know whether we actually found our subventral gland regulator that's regulating effector production in the submental gland cells. And to test this function, we knocked down this transcription factor via RNAi. And then we looked at what happens to gene expression. And in this volcano plot, you can see the results. All of the different dots that you see here represent a different gene. And on the x-axis, you can see the log-fold change in gene expression. And then on the y-axis, you can see significance. So if you focus on all of these dots that are shifted to the left side here, those are the genes that are downregulated if the transcription factor is knocked down. So in other words, this means that this is a positive regulator of these genes because it's required for the expression. And if we take a closer look at this plot, we notice there's a lot happening on this left side. So this seems to be predominantly a positive regulator of gene expression. And we also notice a lot of these circles that are highlighted in blue. And these circles actually represent effector genes. So what this plot shows us, we did indeed find a positive regulator of a lot of effector genes, exactly what we wanted to identify. And that's why from now on, we're going to call this transcription factor the subventral gland regulator short sugar. So now we have our subventral gland regulator, and we know it regulates effectors. So the next question we ask ourselves is, what kind of effectors does it regulate? And when are they important, as it's a subventral gland regulator? And to visualize that, we use this effector network that you see here. In this network, all of the different dots that you see represent, again, an effector gene. And if they're connected by a line, that means that their expression is correlated over the different life stages of the nematode. So genes, effectors that are expressed at the same time tend to cluster closely together here. And we can kind of follow along the nematode life cycle. So if we start at the bottom left, we have J2s that are about to invade the plant. Then if we go further up, we are 10 hours or 40 hours post-infection. And then if we go to the right, we are at the later biotrophic life stages. And now that we can visualize when effectors are expressed, you can add in the effectors that are regulated by sugar. And if we do this, we notice that they're nicely clustered together here at the beginning of the life cycle. So these are effectors that are expressed early on when the nematode has to invade its host. So the next logical question we ask is, how important is this actually for plant invasion? And to test this, we once again knocked down this transcription factor via RNAi, but this time we looked at the phenotype of the number of nematodes that make it into the host. And if we knock down sugar, we can reduce the number of nematodes that were able to successfully invade the host by 80%. So this seems to be highly important early on during plant invasion. And with this, we can kind of start building this model here. We see that root extract activates transcription of our subventral gland regulator sugar, which then orchestrates the production of these early stage effectors that the nematode needs to infect its host. So we start now getting a better feeling of what's happening early on during nematode parasitism. But if we take a closer look at this model, we see there's still a lot missing on this left side. It still looks quite empty here. We wanted to learn a little bit more about this activation of nematode parasitism by this root extract. And the first question we asked is this some kind of generic property of plant extract? Or is this something that's specific to the host of this nematode? And to test this, we had a very similar experiment to what you saw in the beginning, but this time we added in more species. So we still kept mustard, our host plant. We added in another host plant, Arabidopsis, and we added in 2 non-hosts, tomato and rice. And we once again cut off the roots, mash them up, and treat nematodes with this extract and look at what happens to gene expression. And if we do this, we notice that the extract of both host plants was able to activate sugar gene expression, but not the extract of the non-hosts. So at least for the plant species tested, this seems to be actually a property that's specific to the host plants, and the nematode seems to be able to differentiate between hosts and non-hosts. So now that we know that, we got excited and we wanted to learn more. And we wanted to learn more about what kind of signal might trigger this. We started with some very basic crude experiments. For example, we removed bigger molecules, above 3 kilodaltons from the extract, or we boiled the extract to see what happens. And no matter what we did, we still got the same activating effect. So we're probably dealing with a small molecule signal here that's also heat resistant. And another experiment we did is we used columns to remove either strong anions or cations from the extract. And this time, in both cases, we actually saw a slight decrease in the activating effect. So maybe we're dealing with one or multiple charge signals here. So we really wanted to narrow down now whether we are dealing with one signal or but this indicates that there might be actually multiple signals involved. And to test this, we now send the extract to fractionation by HPLC, but all of the molecules inside the extract get separated based on their solubility in aqueous or organic solution. And now we were able to test the activating effect of these different extract fractions that contain different molecules of the extract. And what we notice is that indeed there are multiple fractions that were able to activate sugar gene expression, so we're dealing with multiple signals. And they tend to cluster together in these 3 peaks here. So this tells us we're probably dealing with at least 3 discrete plant signals. And we termed this type of plant signal that activates effector production in the nematode effector stimulants. So now we know effector stimulants exist and they're multiple. The next question we wanted to ask is this something we might be able to breed against and use in plant breeding? Is this maybe something that varies in natural populations and we can select against? And to test this, we asked a mustard breeder for different varieties and tested the extract of these different mustard varieties. We did indeed see some differences. There were 2 varieties that were able to particularly well activate sugar gene expression. And we did the same thing with the other host plant we tested, Arabidopsis, where we saw even more variation. So yes, indeed, effector stimulant production varies in natural populations. And the nice thing about these Arabidopsis thaliana accessions that you see here is that they're actually part of the founder accessions of a set of over 500 recombinant inbred lines, the magic lines, that are all sequenced. And that makes it a really nice resource to actually conduct the GWAS to try to figure out a genomic loci linked to effector stimulant biosynthesis. So that's exactly what we attempted next. So we chose a set of over 100 Arabidopsis magic lines that are all sequenced, and we phenotyped them for their ability to stimulate sugar production, sugar expression. And we once again saw this variation from some accessions that were barely able to stimulate sugar expression to some that were really high inducers. And now that we had this, we could actually conduct the GWAS. And when we did this, we did indeed were successful to identify a significant SNP that's linked to effector stimulant biosynthesis. That SNP is located on chromosome 4, and it's linked to a region that's about 23 kb long and contains 6 genes. So we're currently down to 6 candidate genes that might be involved in effector stimulant biosynthesis. We haven't gotten the chance to investigate them yet, but we are very hopeful and very excited about figuring out the exact and precise molecules that might trigger nematode parasitism. But with this, we can already add in more information in our network here. And we now know that these signals that activate sugar gene expression, nematode parasitism, are small molecule, heat-resistant molecules. There are many, or at least 3, and we term them effector stimulants, and they vary in natural populations. And hopefully we're closer to actually finding them. So now we have a pretty good understanding about these early steps. And we know from the signal to the regulator how effector production is regulated. So the one last question I want to explore on this talk here today is, what about the next wave of effectors? What comes next? Is sugar working alone, or are there actually multiple or different regulators involved? And to find such a regulator of the next stages, we started with what's out there, what's similar to sugar. Sugar is a nuclear hormone receptor, so a type of transcription factor that's often bound by ligands. So we constructed a phylogenetic tree of different nuclear hormone receptors in Heterodera. And you can see the location of sugar here indicated by this red arrow. And we can zoom in into this region. When we do this, we notice that sugar actually nicely clusters together with other nuclear hormone receptors that are highly correlated to effector gene expression. And there's one very close homologue here, HSC gene 17966, that we chose to investigate further. And we wanted to know whether this very, very close gene is another regulator of effectors. We wanted to know what it does. And to explore its function compared to sugar, let's take a look back at what we already know. So this is the network that you've already seen before, where you see the different effectors. We see the sugar activated effectors highlighted in blue here. We see that clustered together at the beginning of the life cycle. And now we wanted to know whether the sugar homolog does the same thing. To test this, we again knocked it down by RNAi, and you can see here another volcano plot that shows you what happens to gene expression if we knock down the sugar homolog. And now you have the differentially expressed effectors highlighted in red here. And if you look at it, we see indeed we did find another regulator of effector genes, but it looks a little bit different to sugar. And the crucial difference here is that this seems to be a positive and a negative regulator of effectors. So you see them on the left and on the right side. And with this in mind, we wanted to know what kind of effectors are repressed and what kind of effectors are activated. So let's add them in the network. Let's add in first the repressed effectors. And if we do this, we notice these effectors that are repressed by the sugar homolog actually nicely overlap with these effectors that were previously activated by sugar. So this homolog seems to switch off the early stage of effectors that were previously activated. We now add in the activated effectors. We see they're clustered together a little bit later in the nematode life cycle. So it seems like we found a switch. It switches off early stage effectors and switches on the next wave of effectors. And this is actually a function that is less suitable for a subventral gland regulator. It sounds kind of like a dorsal gland regulator. So we wanted to know, did we find our dorsal gland regulator? For that, we invested again spatial expression with the same method as we saw before. And you have again in the upper panel now the transcript, this time of the homolog and not of sugar. And below that, again, our dorsal gland control in yellow and our subventral gland control in magenta. And if you compare them, we notice indeed the homolog of sugar nicely matches now our dorsal gland control. So it's specifically expressed in the dorsal gland cell. And that's why this sugar homolog is from now on called the dorsal gland regulator, short DGR1. And with this, we can extend our network. We saw that effector stimulants trigger sugar expression, which then orchestrates early stage effectors. And now we can add in our dorsal gland regulator, which switches off these effectors that were previously activated and switches on the next wave of effectors. So we found our first two regulators of effectors in plant parasitic nematode. Actually, if we take these two together, they control the expression of nearly half of all effectors expressed 48 hours post-infection. So in the first half of the nematode life cycle, or not half, but of the network we saw. So we can really nicely explain the beginning of parasitism in this nematode. And yeah, we're only at the beginning. There's a lot more that we want to explore. A big focus in the future is going to be to actually identify these effector stimulants. We want to find these signals. Another big area that I'm very excited about getting to in future is we would actually like to explore how these effector stimulants are perceived by the nematode, because it might also be a nice idea to target perception. And another area I'm trying to explore is to actually screen for small molecules that might interfere with sugar function, as it's a nuclear hormone receptor. It's maybe a druggable transcription factor. And with all of these things in common, the principal idea is to target a central regulator of nematode parasitism and thereby take away the key tools that the nematode needs to infect the plant. And with this, I'm at the end of my talk. I want to thank everyone involved in the project, in the group, all the funders, and thank you very much for listening.